Today I'm going to be talking about an example of developmental networks. And our example today is going to be the thorny dragon lizard. The specific pathway we'll be looking at determines whether or not the lizard will have spikes on it. So we have multiple genes here. We have the S gene promoting the P gene inhibiting the I gene inhibiting the K gene, and the K gene promoting the E gene, which gives us spikes. Now, all of these all together in the wild type gives us spikes because S promotes the P keeps the I from inhibiting K by inhibiting it, so the K can promote E, which gives spikes. So let's say we didn't have a P gene. Without the P gene, that means I is no longer inhibited and can inhibit K. And if K is inhibited, that means E cannot be promoted and spikes will not occur. So what would happen if we didn't have an I gene in our pathway? So without the I gene, that means we are no longer inhibiting K, which means K can promote E, which can give us the spikes. So we would have a spiked lizard. Now what if we were missing both the I and the E? Now as we looked at in the previous slide, if we only didn't have the I gene, we would have spikes because K would not be inhibited and E would then be able to be promoted, which would give us spikes. But we also don't have E, and since E is our last step to give us the spikes, we do not have spikes and we have a smooth lizard. So what would happen to our pathway if there was no S or K gene? So without an S gene, that means P would not be made and I would not be inhibited, which would inhibit K, which would mean no spikes. And inhibiting K would be the same as if we didn't have it, and in this situation we don't have it, so this would be no spikes. So we look at the lizard here and we see that not every little cell, every little bit of his body is covered in spikes, which means there must be some sort of selective regulation. But how can that be if all the cells have the same genetic code? I hypothesize that the pathway is controlled by a positive inducible regulatory method on the S gene. So that means just that the RNA polymerase alone will not create a gene for the S gene. It needs an activator. However, with just the activator, we still can't translate our gene. We need our small molecule right here. And what this will do is this will activate our activator, and this will allow our RNA polymerase to translate the gene.